Hi everyone, this is the lab recording on microscopy. So there are two main types of microscopes. There are light microscopes, which use visible light to illuminate specimens, and that's what we typically use in our teaching laboratories and what you would typically come in contact with in any type of a medical lab setting. Then there are electron microscopes, and these use electron beams instead of light rays, and they use magnets instead of lenses, and we're able to see very fine detail on a much higher magnification level than microscopes. So you'll only see electron microscopes in research labs, maybe in very big hospitals, like the Medical University of South Carolina or someplace like that, where they would have the funds for one of these because they're very expensive. We're interested in this type of microscope. There are lots of variations to these based on the manufacturer, but they all have sort of the same stuff. So we're going to start up at the top left-hand side, and we're just going to go down, and I'm going to point out each of these different parts of the microscope and what each one does, and that's what you would want to know. So we'll start with the ocular lenses, which is up at the very top. Sometimes referred to as the eyepieces. They are a standard of 10x magnification, and you can determine that for sure by looking at the side, though it'll be stamped on the side. There are greater magnification oculars, but the standards that most people use are 10x. Going straight down from there, we have the part that moves around. It's called the rotating nose piece, and from that rotating nose piece are the tubes that we call the objectives. This allows you to change from one magnification to the next. And then you have the objectives. The standard objectives that we use and that we're going to learn about today are 4x, 10x, 40x, and 100x magnification. There are others. You can again look at the side and it will tell you the magnification. You can special order all kinds of magnifications, but these are the standard ones that we typically see. Then below that is going to be the mechanical stage. This is what supports the microscope slide during observation. And then underneath of the stage, we have the iris diaphragm, which allows us to adjust the amount of light that's coming through the microscope aperture, the stage aperture, where we can so we can see what's on the slide. Then below that, you have the condenser, and that's what focuses the beam of light. It condenses the beam of light onto our specimen. We have our light source down there on the bottom, and then we have the base, which is what is in contact with the lab bench. Going from the top over on the other side, we have the binocular head, which is where we have our two eyepieces in this particular case and our prisms. That's the really expensive part of the microscope. Then you have the arm, which is what connects the head to the base and what we carry the microscope with. And then we have the X and Y knobs, sometimes referred to as the stage adjustment knobs. These are what move our slide around when we're trying to find where it is on the specimen on our slide. Then we have our coarse adjustment and our fine adjustment knobs. And so the coarse adjustment knob is the larger one, and it moves the stage up and down, and you can actually visibly see that happen. And we use that for scanning in low power. Then we have the fine adjustment knob, which is nested inside of that. And so this is when you move that, you can't really determine if the stage is moving if you're looking at it, but if you're looking through the microscope, you can see that it's moving just a little tiny bit. And we use that one under our higher power lenses. And then finally, you have the light switches, which is what turns the microscope on and off. So taking a little bit closer look at that coarse adjustment knob and the fine adjustment knob, how those are two nested together. Sometimes they're separate. Again, it just depends on the manufacturer, but the coarse adjustment knob is going to move the slide up and the stage up and down And we use that on the first two objectives, what we're going to talk about in a few minutes, the scanning and the low power. And then the fine adjustment knob we use to get finer detail and to get it in fine focus. And that typically is what we're using when we're on the higher magnification objectives. Over on the right, they're showing you the condenser knob, and that's what raises and lowers the substage condenser. Typically, we ask students not to touch that because it's real easy for that to get messed up. So we just say it's already been set. We have somebody who comes through and checks all of our microscopes, and it's already been set. So we ask you to just leave that alone typically. And then the dimmer knob, this one's really important. This is a rheostat. It's like if you had a dimmer switch in your house. It adjusts the light intensity from the light source. Sometimes it's a slide, sometimes it's a knob. It's just kind of variable. If you ever look through the microscope and it is so bright you're seeing stars, you want to immediately try to see if you can get that dimmer to go down just a little tiny bit. And then just a drawing. This is a monocular lens, a monocular scope that we're looking at here. It appears to be at least. And it's showing you the pathway of light. So how the light goes through the microscope and allows you to see it. So obviously the light comes from down at the base where it is, and it's going to go up into the condenser. And the condenser is going to condense it through the aperture. There's a little hole in the slide or in the stage, and the light's going to come right through that, and then it's going to go up into the objective lenses and through all the prisms, and then ultimately the pathway of light is going to travel to the oculars where you're able to see it. So what we need to look at now is the how much magnification that we are using when we use the different objectives and the oculars. So remember, the oculars are 10x. We're going to assume 10x here. All right. And so the formula for calculating total magnification is always going to be the ocular times the objective magnification. All right. And so that's pretty straightforward. We're going to start with the scanning objective, which is the shortest one. Its magnification is 4x. We're going to multiply that by the ocular, which is 10x. And so our total magnification, how much bigger we're making it by using the scanning objective, is going to be 40x. When we look at the low power objective, its magnification is 10x. We're going to multiply that by 10 for a total magnification of 100. When we look at the high power, its magnification is 40x. We're going to multiply it by 10 to get a total magnification of 40x. And then finally, our oil immersion lens, which has a magnification of 10x. We multiply it by 10 to get a total mag of 1000x. So you should be writing it like you see on here. The number with a small x, no space between them, anything like that. So let's look at a little bit of microscopic views of what we can see. All right, so we're looking through the microscope, and we can see over on the left-hand side we have a gram-negative stain, and we'll know why that's gram-negative in just a couple of weeks. And you can determine the shape and the color and all that sort of thing. And then on the right you see a gram-positive stain. Down at the bottom you see some streptobacilli that are gram-positive as well. We're able to compare our eukaryote and our prokaryotic cells. So these great big ones, the big things you see that look kind of like a fried egg, those are squamous cells. You should have seen those last semester or first semester in anatomy and physiology. Those are simple squamous epithelial cells. And then the little teeny tiny debris that you see everywhere, that's a bacteria. So it really gives you the idea of the difference in size that we have between our eukaryotic cells, which you have observed in anatomy class, and the bacterial cells. So when we look at the bacteria underneath of a microscope, showing you the power of these different lenses. So when we're on 40x, which is the scanning, the low power objective, you can't really see a whole lot. You can see color. You might be able to see some shape, but you definitely can see some color. Under the low power, the next one down, you see a little bit more detail. When we get to the 400x, you really start to see the shape and you can discern a lot more detail now. And then finally, under oil immersion, which is the 100x, you'll be able to see a lot more finer detail, which is why we use the oil immersion lens. Here's the same idea. Bacteria underneath the microscope, 40 to 1000x. So how do we use a microscope? Again, you're in a digital lab, so you're not going to be doing this, and you may have used microscopes in a previous course that you took, but you certainly will see them out in labs if you work in a doctor's office. So be a microscope there that you can do some simple lab work with. And then, of course, if you were to work in a hospital lab, you would use them all the time. All right, so you're going to place the microscope slide on the stage with the stage clips. That's the first thing you're going to do. You're going to handle the microscope slide with the edges. You don't want to put your fingers on the middle of the microscope slide. Then you're going to raise the stage up completely. You always leave the microscope with the stage completely down. So when you're finished with your microscope, you leave it down. So the next person that's going to come along is going to slide their slide in and then raise it straight up. Then you're going to center the specimen over the light source. You're going to turn your light on, obviously. You're going to rotate the nose piece and get yourself to the scanning objective. And while looking through the ocular lens, you're going to use the coarse adjustment knob, which was the larger of the two, to focus on the specimen. Then you're going to rotate. Once you got it all nice and focused, you're going to rotate over to the low power 10x objective. And while looking through that, you're going to, again, use the coarse objective knob as well as the fine objective knob if you need to get it in better focus. Then you're going to move over to that high power 40x objective. Now you are only going to use the fine adjustment knob. You never use the coarse adjustment knob after you get past that 10x because you could potentially break the slide or even worse, break the objective because the two are really close to each other. So the fine adjustment knob only once you get to the 40x or above, and then you're going to move that out of the way, and then you're going to do what's called oil immersion. So we only do oil immersion in microbiology. We do not do oil immersion in anatomy and physiology. Oil immersion allows us to see detail at much finer magnification, much higher magnification. So we only do this because you just saw how tiny bacteria are compared to a prokaryotic cell. So it's the only time that we do this. So if you were in a teaching lab on campus for your anatomy and physiology, they don't even have that 100x objective on the microscope. They take them out so you don't use them, right? So how do you use them? You get everything in focus the way you want it, and then you're going to get your take your 40x and you're going to move it over to the side. That's the purpose of that rotating nose piece. Then you're going to put a drop of oil specifically used for this called oil immersion oil. You don't use anything else. You don't find anything you happen to have around. You use oil immersion oil for this. You put a drop of it on the slide where you're looking at, and then you swing that nose, you swing the objective, the oil immersion objective, into place on top of that oil. All right. When you are looking at the specimen under the 100x, you can use the fine adjustment just a tiny amount if you need to get more clarification, more clarity in your vision that you're looking at there. So here's what I was just talking about, using that oil immersion lens. It gives us greater clarity, greater resolution when we use the oil immersion, and the objective requires the use of this oil to prevent refractive light loss. So even though when we're at 100x objective and we look at the space between the objective and the slide, it's just like a hairline space, but light can still scatter. And so if we use that oil, it keeps the light from scattering. And so we can see greater detail. And so that's the whole point of using the oil immersion lens, is that we can see greater detail because the light doesn't scatter. Once the oil is on the slide, you do not sweep back to the 40x objective If you do that, you need to make sure you need to make sure that you clean all of the objectives. That oil will cloud the objective if it's left on there. So this is why we don't use it in our other classes, because it's just one more thing to try to manage. And since you don't really need it, is that someone would accidentally somehow or another use the oil immersion lens with oil. So we just make sure they're not on there. If you get oil on any other objective, you want to get it off immediately. After you're finished observing your specimen, you're going to clean the lenses with lens cleaner and lens paper, and then we're going to do the oil immersion lens last because we don't want to transfer the oil back to the other objectives. We're going to wipe off the specimen slide, and then we're probably going to reuse that specimen slide unless it's one that you made and it may go into the glass breakable box. Put the 40X, which is the red ringed, the low power, the 4X objective back in place on the rotating nose piece. Wrap the cord around the base, put the cover back on the microscope, and put the whole thing back in its correctly numbered slot. Obviously, if you're in your own lab somewhere, you wouldn't go to that last part, but you would cover the microscope. And I can't say this enough about covering microscopes. Don't leave microscopes uncovered for any length of time. They collect dust like a magnet. And so it won't take very long before you're not getting good, uh, accurate viewing because of the dust. So even in a laboratory, if you leave for the day, you want to make sure that you cover your microscope with its microscope cover. That will prevent dust from settling. You have some labs through Connect that you'll want to complete. So make sure that you do all of those. And if you have any questions, please let me know, and I'll see you for the next lab.